Royals, Red Hair, and Dürer, Part 1. Copyright February 15, 2014, by Dr. Elizabeth Garner and Joe Kiernan. Biological protection of red hair. Natural red hair is very uncommon within the total gene pool of humans. DNA research regarding the existence of natural red hair has uncovered that there is a biological advantage to having red hair in areas of the world where red hair occurs statistically above the norm. Scientists have found that ginger or red hair and a pale skin offer an important advantage in the survival game. Redheads, it would seem, boast a secret genetic weapon which enables them to fight off certain debilitating and potentially deadly illnesses more efficiently than blondes or brunettes. A pale complexion permits more sunlight into the skin, where it encourages the production of vitamin D. This helps to prevent rickets, a disease which progressively weakens bone structures, and the lung disease tuberculosis, which can be fatal. A recent study in the US suggested that people with red hair are more sensitive to pain than blondes and brunettes. MC1R and R1A1, basically the same thing. The gene that determines red hair is MC1R, which used to be referred to as R1A1. Researchers consider strawberry blonde and auburn hair to be different from real or fiery red hair. About 90% of the time, it is indeed having two copies of variant MC1R that leads to having fiery red hair. However, there are a number of different variants of MC1R, only some of which lead to having red hair. One variant, for example, seems to lead to having fair, blonde or light brown hair. Three variants of MC1R known as allele seem to strongly promote having red hair, and having two copies of these variants in any combination almost guarantees having fiery red hair. If a person has only one copy or no copies of the red hair variants, that usually means that the person will have either non-red hair, strawberry blonde hair, or auburn hair. That fits with the classic model of a recessive trait. However, if a person has one of these strong variants plus the variant for fair hair, red hair acts as a dominant trait. The real differences between males and females. The Y chromosome and the X chromosome characteristics. Geneticists study genes differently depending upon whether they are studying the Y chromosome, which makes males, and the X chromosome, which comes from the mother and is referred to as the mitochondrial DNA. Mitochondrial DNA tells us much different stories. There, of course, have been enormous advancements in the studies of genetics and paleogenetics in the last decade, and therefore, geneticists started needing to rename the whole classification. Ultimately, what is now called the red hair MC1R gene used to be called the R1A1 haploid group. The historic naming system commonly used for the R1A was inconsistent in different published sources, and there are now very complicated conversion tables about these renaming conventions. For our purposes, we really need to only concern ourselves with the fact that it is within the R1A1 haploid group that natural red hair occurs genetically. We know that the British Isles have the highest concentrations of redheads, and thus R1A1 is much higher statistically in those populations, but most people don't know that the highest other concentrations of redheads came from Persia. Persia covered an enormous area of land historically where the natural red hair occurred, and we get complicated again. Besides modern Iran, Persia, ethnic Persians are also found in Central Asia, in Afghanistan, Tajikistan, Uzbekistan, where they are usually called Tajiks, and Phariswans, as well as in southern Iraq, Babylonia, a region which has been historically an integral part of Persia. Some names such as Tat, Tajik, Sart, and Ajem have also been used, especially by the Arabs and the Turks, in reference to the Persians. The terms Parsi and Tajik and Tat have been used interchangeably for Persian and Persian speakers during the Middle Ages. As it turns out, Jews have been enormously interested in tracing their genetic heritage, and there is much data about the R1A and the R1B haploid groups that has been gathered and published. Much of this research focuses on the ethnic sources of Jews in the Persian area, whether distinctions of Kohanim and Levites, two important Jewish priestly classes, could be determined, and how much of the evidence told us about the mysterious Khazars, known for their red hair, the only entire country that formally converted to Judaism in all of history, roughly 600 to 900 AD. The Khazars are the most likely ancestors of the Durers, since the father emigrated to Nuremberg from the second biggest Khazar area ever found. The Dürer family originates from one of the major Khazar regions formerly known as Bihar or Grosswardine, and now Ordia in the Romanian part of what used to be the medieval Wallachia, next to Transylvania of Dracula, Vlad Tepes the Impaler. Albrecht and Margaret had red hair. Who's your dad? Mutations which occur within genes usually cause a malfunction or disease and is lost due to selection in succeeding generations. However, mutations found in so-called non-coding regions of the DNA tend to persist. Since the male Y chromosome consists almost entirely of non-coding DNA, except for the genes determining maleness, it would tend to accumulate mutations, from father to son without recombination. Thus, the genetic information on a Y chromosome of a man living today is basically the same as that of his ancient male ancestors, except for the rare mutations that occur along the hereditary line. A combination of these neutral mutations, known as a halotype, can serve as a genetic signature of a man's male ancestry. Maternal genealogies are also being studied by means of the mDNA, mitochondrial DNA, which is inherited only from the mother of the egg. Jewish priests. Judaism is known as the Kohanim. The Kohanim, plural of Cohen, are the priestly family of the Jewish people, members of the tribe of Levi. The books of Exodus and Leviticus describe the responsibilities of the Kohanim, which include the temple service and the blessing of the people. Aaron, the brother of Moses, was the first high priest, Cohen Gadol. The Cohen line is patrilineal, passed from father to son without interruption for 3,300 years, or more than 100 generations. The results of the analysis of the Y chromosome markers of the Kohanim and the non-Kohanim were indeed significant. A particular marker, YAP, was detected in 98.5% of the Kohanim and in a significantly lower percentage of the non-Kohanim. This was called the CMH group. The finding of a common set of genetic markers in both the Ashkenazi, the Jews from Germany and Eastern Europe, and the Sephardi Kohanim, the Jews from the Iberian Peninsula, Spain and Portugal, worldwide clearly indicates an origin predating the separate development of the two communities around 1000 CE. Date calculations based on the variation of the mutations among the Kohanim today yields a time frame of 106 generations from the ancestral founder of the line, some 3,300 years, the approximate time of the exodus from Egypt, the lifetime of Aaron HaKohen, the name given by scientists to the supposed progenitor. Infidelity. Calculations based on the high rate of genetic similarity of today's coenym everywhere resulted in the highest paternity certainty rate ever recorded in popular genetic studies, a scientific testimony to family faithfulness. Dr. David Goldstein of Oxford University stated, "For more than 90% of the Cohens to share the same genetic markers after such a period of time is a testament to the devotion of the wives of the Cohens over the years. Even a low rate of infidelity would have dramatically lowered the percentage." The Levite priest class. The DNA of the Jewish Khazarian priests. The other Jewish priestly caste is known as the Levites. Like the Kohanim, Levites are recorded in the Hebrew Bible as direct descendants of Aaron, Israel's first high priest. In fact, the Kohanim are actually a special subsection of the Levites. The Levites were shown to have a common set of genetic markers, just not the CMH set of the Kohanim. These markers were not even part of the same J1 haploid group as found in the Kohanim. The majority of Levites share a common haplotype, indicating a shared common ancestor among them. But this haplotype occurred within the haplogroup R1A1 and, more specifically, within subgroup R1A1, red hair. Furthermore, this haplogroup was found only in the Ashkenazi Levites. It was not shared with the Sephardic Levite population in the same fashion as the CMH. Given the fact that the Ashkenazi Levites did not share R1A with the Sephardic counterparts, it appeared that this haplogroup had entered the Jewish population sometime during the diaspora. Researchers found that R1A1 formed a tight cluster within the Ashkenazi Levites. This suggested to the researchers a very recent origin of this group from a single common ancestor. We know from subsequent research that R1A1 comprises nearly 12% of Ashkenazi results, while the Levites only make up about 4 to 5% of the entire world Jewish population. Thus, these results extend well beyond the Levite priestly class to approximately 5 to 8% of the Kohanim and Israelites, the non-priestly Jewish population as well. Haplogroup R1A1 is relatively rare within the Middle Eastern populations, but very common among Eastern European and Scandinavian populations. It is found a frequency of 7% of some Near Eastern groups. However, given that Sephardic Jewish Iberian groups did not share the R1A1 frequencies with the Ashkenazim, it was apparent that the Jewish R1A1 was probably not of ancient Israelite origin. In other words, red hair was not common in the Middle East. R1A1 is spread fairly even in a haplotype distribution and frequency throughout the Ashkenazi populations from various countries: Germany, Lithuania, Czechoslovakia, Hungary, Romania, Poland, Russia, and the Ukraine. The Jewish Khazar Nation. The mysterious Khazar nation is the only group of people known in the entire history of the world to have converted to Judaism and to have required their top nobles to do so. Lesser nobles and non-nobles were allowed to maintain any religion they wanted, Jewish, Christian, or Muslim. They were known from the small amount of historical documents that survived and to have had flaming red hair. There are no historical accounts of any large-scale conversions or Eastern European groups entering the Jewish community at this time, except the Khazars. Additionally, given the relatively late date of integration and the large number of founders, these males must have already been very closely related to each other, sharing the R1A1 haploid types that are later reflected in the Levite results. Baer in 2003 noted that the lack of Levite R1A1 haploid diversity suggested that all the founding lineages were very closely related to each other if, in fact, a large number of founding lineages contributed to the Levite R1A1 gene pool. The ancient reports on the Khazars indicate that the majority of the Jewish converts were from the Khazarian royalty and ruling classes. Although speculative, it seems likely that this group would have intermarried heavily amongst itself, helping to preserve the group's elite status. Thus, it is probable that they would have already possessed a set of closely related R1A1 haploid types, which they simply passed on to their Levite descendants. The Khazars were already Jewish, having converted around 600 CE, although of a different ethnic makeup than that of the Ashkenazim of the 13th century. They were not non-Jews. They probably already had their own Levite caste in place, who may have simply continued their priestly functions among the Ashkenazim. As for when the R1A1 red hair gene first entered the Jewish community, Baer estimated a mean time of 663 years before the present. This calculation was striking because it fit precisely within the time period that Kostler believed the mass migration and absorption of the Khazars by the larger Eastern European Jewish communities occurred. In other words, when the Khazar nation was conquered and absorbed by the other groups. Jewish red hair genes are the same as Christian red hair genes. R1A1 is found in very high frequencies, not only in the area of Eastern Europe where the Khazarian kingdom is reportedly to have existed, but also in many Central Asian populations as well, where some of the Khazarian populations may have originated. Furthermore, the most common Ashkenazi haploid type, H6, is identical to the most common haploid type found among European R1A1. Ashkenazi H10 is identical to the fifth most common European R1H1 haploid type. In other words, Jewish red hair genes are identical to Christian red hair genes, as they are dispersed around Europe.